Hi, everyone. This is Jason Key from SBGrid in Boston, Massachusetts. Thanks for joining today. Just want to kick off with a couple of announcements. So our webinar series is in full swing. So we are going straight through the fall. We've got about one per week. So October's lineup, just rounding it out. We've got Lance Westerhoff coming up next week. He's going to talk about movable type for fast, free energy-based virtual screening. Then we're going to talk about vaccines with Sophie Gobiel from the HR group at Duke. Then October 20th, Jose Miguel de la Rosa Trevin is going to talk about Scipion 3, which is sort of big Cryo-EM processing package that includes all kinds of different workflows for processing Cryo-EM. So I'm excited to see what's coming in version 3. We've got another one for the 27th that's still open. And check our website, sbgrid.org. We've got all of them listed there. We've got talks running, clear through December now. So be sure to join us for that. With that, it's my pleasure to introduce Qing Fan from Columbia University. So Qing's going to be telling us today about the cryo-EM structure of human GABAB receptor. So go ahead and if you could share your screen, you should be able to take over the screen there.
Okay, thank you.
And as we go, just feel free to send me questions by chat. And at the end, we'll have time for more questions. We can unmute and hopefully have a little discussion as we go. So, and if you could go ahead and full screen, it should, looks great. Go right ahead.
Okay, first, I want to thank SBGrid for inviting me to present our work. And today I will talk about the Cryo-EM structure of human GABAB receptor. As you will see, this is really teamwork, and we collaborated with multiple groups to obtain the results that I'm about to show you. Specifically, we worked with Dr. Joachim Frank, his student Zhao Fu, and Dr. Oliver Clark on the Cryo-EM method. We also worked with Dr. Oliver Finn and Dr. Tongshen on lipid identification, and Dr. Joseph Graziano and Dr. Vesna Slakovic on elemental analysis. The function of human brain involves both excitatory and inhibitory signals. Inhibitory neurotransmission is primarily mediated by GABA. GABA activates metabotropic GABAB receptor, a G protein coupled receptor that is widely distributed in the central nervous system. It produces slow and prolonged inhibitory activity. GABAB receptor was initially discovered on the sympathetic neuron terminals by Dr. Norman Bowery, and biochemical isolation of the receptor was first achieved by Dr. Bernard Bettler's lab. Malfunction of GABAB receptor has been implicated in a number of neurological diseases. Baclofen, a selective agonist of GABAB receptor, is used clinically to treat muscle spasticity in patients with multiple sclerosis and spinal cord injury. GABAB receptor belongs to the subfamily of class C G protein coupled receptors, which also include the metabotropic glutamate receptors, calcium sensing receptor, and sweet and umami taste receptors. GPCRs are divided into 4 main classes, A, B, C, and F, based on the sequence homology of their transmembrane domains. Most GPCRs, such as rhodopsin and beta-2 adrenergic receptor, belong to class A. They contain a 7-helix transmembrane domain and can function as monomers. The ligand binding site of the class A receptors is located within the transmembrane domain. Studies led by Dr. Brian Kavilka's group showed that agonist binding directly induces conformational changes among the transmembrane helices for receptor activation. Unlike the class A receptors, the class C GPCRs are characterized by a large extracellular domain, in addition to the canonical 7-helix transmembrane domain. This extracellular domain is 500 to 600 amino acids long and contains the orthosteric agonist binding site. Another unique feature of the class C receptors is that they require dimerization for function. The metabotropic glutamate and calcium sensing receptors function as homodimers. The GABAB and taste receptors function as heterodimers. GABAB receptor functions as a heterodimeric assembly of the GABAB1 and GABAB2 subunits. This unique functional property of GABAB receptor was discovered in 1997 by several groups simultaneously. The two subunits are structurally homologous, but perform distinct and complementary functions. GABAB1 is responsible for orthosteric ligand binding through an extracellular venous flytrap module. GABAB2 facilitates the cell surface expression of GABAB1 and is involved in G-protein coupling. Our goal is to understand the signature of GABAB receptor using structural methods. Previously, we solved crystal structures of the extracellular VFT module of GABAB receptor in multiple functional states. As shown here is the GABAB VFT module in the apolyl form, bound to an antagonist and bound to an agonist. Each subunit has a venous flytrap-like architecture and consists of the LB1 and LB2 domains. Both the agonist and antagonist bound in the crevice between the LB1 and LB2 domains of GABAB1 subunit. Within each receptor complex, the two subunits bind to each other in a side-by-side fashion, but face opposite directions. The dimer arrangement looks like Fred Astaire and Ginger Rogers dancing cheek to cheek. The apolyl and antagonist bound structures closely resemble one another. Both subunits adopt an open conformation within these complexes. The open-open configuration corresponds to the inactive state of the receptor. Agonist binding causes the extracellular domain of GABAB1 to close. The LB1 and LB2 domains clamp down on the agonist, just like what a venous flytrap does when a fly enters its groove. On the other hand, the GABAB2 subunit stays open. This closed open configuration corresponds to the active state of the receptor. This animation illustrates the conformational changes associated with agonist binding. First, agonist induces domain closure of GABAB1. Second, GABAB2 remains open, indicating that it has a constitutively open conformation. Third, the LB2 domains of both subunits undergo a twist motion such that they come into close proximity to form a large heterodimer interface that's unique to the active state. In addition, agonist binding shortens the distance between the C-termini of the two subunits. The decrease in the separation between the membrane proximal LB2 domains is expected to be sensed by their transmembrane domains for receptor activation. Recently, we solved the structure of a near full-length GABAB receptor using Cryo-EM in collaboration with Dr. Qing Fan and Dr. Oliver Clark. Three other groups led by Drs. Cherezov and Gatti, Drs. Zhang and Liu, as well as Dr. Skiniotis, also published similar structures at the same time as we did. In our case, we expressed the human receptor in baculovirus-infected mammalian cells. For each subunit, we used an expression construct that was truncated at the C-terminal end according to the domain boundary of the intracellular coil-to-coil. This strategy allowed us to remove the flexible region in the cytoplasmic tail. The coil-to-coil fragments also promote heterodimerization and facilitate cell surface expression of both receptor subunits. As shown here, the receptor subunits assembled as a heterodimer upon gel filtration chromatography. The receptor can be extracted from the cell membrane and purified to homogeneity using detergent. Jingseo Park from my lab purified the protein with assistance from Jonathan Liu on cell culture and Kim Ray on membrane preparation. Our collaborator, Dr. Matthias Quick, measured the binding of radioactive GABA to the recombinant receptor. We found that the GABA affinity for the receptor was comparable to that of native receptors. This indicates that the detergent did not alter its ligand binding property. In addition, we compared the signaling function of the truncated and full-length receptor. Jonathan Liu and Kim Ray from my lab conducted the functional assay. Jingsaw Park and Aurel Frenge made the constructs, and we obtained advice and help from Dr. Paul Schlesinger at Mount Sinai and Dr. Jonathan Javich at Columbia. Briefly, we co-expressed GABAB subunits with a chimeric G-alpha-Q protein, QI protein, which couples the receptor to the GQ signaling pathway and phospholipase C. The function of the GABAB receptor could then be monitored through the accumulation of inositol phosphate. As shown here, application of the GABAB agonist baclofen resulted in dose-dependent IP production. We found that the C-terminally truncated construct respond to baclofen with similar potency and efficacy as the full-length receptor, indicating that the deleted regions in the GABAB1 and GABAB2 cytoplasmic tails are not required for receptor activation. To solve the receptor structure, we tried both crystallography and Cryo-EM. Unfortunately, we were not able to obtain any diffracting crystals of the receptor. We also struggled with Cryo-EM initially. We obtained help from multiple groups, including Dr. Richard Henderson and Dr. Xiaoxia Chen at MRC, and Dr. Bill Rice and Dr. Ed N at NYSBC. We eventually set up collaboration with Dr. Joachim Frank's group here. His student Zhao Fu showed Aurel Frenge from my lab the entire Cryo-EM workflow, and the help from the Frank lab allowed my group to finally enter the Cryo-EM field, and I will always be grateful. Zhao and Aurel together screened many different protein preparations and eventually identified an optimal freezing condition for this receptor. As shown here is a representative micrograph they collected on a Krios microscope. However, even with this kind of data, we were lingering at 5 to 6 angstrom resolution. Dr. Oliver Clark then came along and applied the non-uniform refinement in CryoSpark that was still in beta testing phase at the time. This strategy, combined with his other approaches, allowed him to reach a global resolution of 3.3 angstrom. And here are his 2D classes and 3D global reconstruction colored according to local resolution. Oliver further improved the map by conducting local refinement of the extracellular and TM domains separately. This is because the global map displayed directional anisotropy, possibly due to flexibility of the linker region. Here are the FSC curves for the global and local refinement. He achieved 3.1 angstrom resolution for the extracellular domain and 3.4 angstrom for the TM domain. Here is a movie of the composite map. It showed clear side chain densities for almost all of the residues. This map allowed us to build a model for the GABAB receptor. We used a crystal structure of the VFT module as the initial model for the receptor extracellular domain, but we were able to trace the linker and TM domain of each subunit de novo, and Lydia Mosiak built the model. As shown here, the heterodimeric GABAB receptor is assembled by GABAB1 and GABAB2 subunits interacting side by side while facing opposite directions. Each subunit consists of an extracellular VFT module, linker, and TM domain. The receptor subunits associate through both the extracellular and TM domains. To our surprise, we observed multiple ligands bound to the receptor in the Cryo-EM density map, even though no ligand was added during expression and purification. These included a calcium ion bound to the extracellular domain of GABAB1 and a large phospholipid within the TM domain of each subunit. We also observed cholesterol molecules around the exterior of the TM domains. Here is a cartoon of our structure. We can see the bound calcium ion, phospholipids, and cholesterol molecules. Within each subunit, the elongated linker interacts extensively with the extracellular loop 2 of the TM domain. And these components together serve as a mechanical junction to transmit the conformational changes in the VFT to the TM region. Oliver also performed 3D variability analysis of the Cryo-EM data and found that the linkers in both GABAB subunits exhibit intrinsic flexibility, since the extracellular and TM domains of the receptor spontaneously flex back and forth about the linker. Based on comparison with the VFT crystal structures, we concluded that we have captured the near full-length receptor in an inactive conformation for two main reasons. First, the extracellular VFT modules of both subunits adopt an open conformation. As shown here is an alignment of our Cryo-EM structure, which is shown in cyan, with the inactive state crystal structure of GABAB VFT shown in purple. It shows that each individual subunit has an open interdomain cleft, while GABAB2 is constitutively open regardless of state. The open conformation of GABAB1 is associated with the inactive state, since agonist typically induces VFT closure. Second, I mentioned earlier that a hallmark of the active state VFT structures is a novel heterodimer interface between LB2 domains, which results from conformational changes induced by agonist binding. If we cross-link the LB2 domains together through a disulfide bond, the receptor displays constitutive activity, indicating that the formation of the LB2-LB2 heterodimer interface is both necessary and sufficient for receptor activation. Indeed, there's a large separation between the membrane proximal LB2 domains of GABAB subunits in our Cryo-EM structure. When viewed from the bottom of the LB2 domains, the distance between the C-termini of the LB2 domains in the Cryo-EM structure is comparable to that of the inactive state VFT crystal structure. The Cryo-EM structure is again shown in cyan, and the VFT crystal structure shown in purple. Therefore, the large separation of the LB2 domains in the Cryo-EM structure is consistent with an inactive conformation of the receptor. Using conformational variability analysis, again done by Oliver, we also observed that the LB2 domains and linkers of both GABAB subunits fluctuate by approaching and withdrawing from the central vertical axis of the heterodimer. However, these domains never come into direct contact, as in the active crystal structure of GABAB VFT. The motion exhibited by the LB2 domains and their associated linkers suggests that the inactive functional state corresponds to an ensemble of conformations, and the separation between the membrane proximal regions falls within the small range around the coordinates of our structure. In the Cryo-EM structure of GABAB receptor, we observed a novel heterodimer interface between the TM helices 5 and 3 of both subunits. This interface represents the signature of GABAB receptor's inactive conformation. These TM helices form a cone and interact at their intracellular ends. Although their extracellular ends do not make contact, they approach and withdraw in an oscillating motion that is part of the conformational variance displayed by the receptor, and this is shown here. This slide shows that the direct heterodimer contacts can be divided into 3 layers along the helical path of TM5. The interactions in the top 2 layers are hydrophobic contacts formed by leucine and phenylalanine residues of the TM5 helices. At the bottom layer, however, we observed a unique set of interactions mediated by a quartet of charged residues. They're from the TM5 and TM3 helices of both subunits. We named this feature the inter-subunit latch for joining 2 subunits of the heterodimeric receptor. In addition, the central layers of direct heterodimer contacts are flanked on each side by a cholesterol molecule. One mediates the interaction between TM5 helices of both subunits, while the other bridges TM3 of GABAB1 and TM5 of GABAB2. We hypothesized that the intersubunit latch is important for controlling the inactive state of GABAB receptor. To investigate this, we examined the effect of single charge repelling mutations within the motif. These include E673R in GABAB1 and H579E in GABAB2. The wild type data is shown in black and the mutant data are shown in red and blue. The lighter shades of color correspond to data obtained in the presence of an inverse agonist. As shown here, wild-type GABAB receptor displayed basal activity in the absence of agonist. Both mutants substantially increased basal activity when compared to wild-type, suggesting that each mutation promotes agonist-independent constitutive activity. The basal activity displayed by these mutants reached approximately 80 to 90% of the maximal agonist-dependent wild-type response, as shown here. An application of the agonist baclofen raised the receptor activity further to the highest wild-type level. After treating each construct with an inverse agonist, the basal activity of the mutants still remained higher than that of wild-type. This observation provides further evidence that the intersubunit latch mutations serve to shift the conformational equilibrium of GABAB receptor towards an active state. Taken together, our mutational data indicate that the intersubunit latch is fundamental to maintaining the inactive state of the receptor. I mentioned earlier that we observed multiple ligands pre-bound to the receptor. First, we identified a novel calcium binding site at the interdomain cleft of GABAB1 VFT, and it is located in the vicinity of the orthosteric ligand binding site. We first recognized the metal ion density by its remarkable peak height in the Cryo-EM density map. In addition, it is surrounded by residues that are chemically favorable for calcium ion coordination. Finally, our collaborators, Dr. Joseph Graziano and Dr. Vesna Slakovic, performed elemental analysis of purified GABAB receptor using inductively coupled plasma mass spectroscopy. They showed that calcium is present in our protein above background levels. We explored the functional role of the bound calcium in two ways. Again, the wild type data is shown in black and the mutant data are in green and cyan. First, we found that the presence of a calcium chelator, EGTA, substantially reduced the basal activity of GABAB receptor. This is shown in gray in both diagrams. Second, we introduced single charge reversal mutations at the calcium binding site, E309K and E423R in GABAB1. Both mutations had similar effects on the basal activity of GABAB receptor as EGTA, but to a lesser extent than EGTA. These data suggest that calcium may act as a positive allosteric modulator of the receptor. And this is consistent with previous findings by Dr. Fiona Marshall and Dr. Jean-Philippe Pen's groups that calcium ion potentiates agonist activity in GABAB receptor. Our structure suggests that the binding of calcium may provide an essential framework for agonist recognition through its effect on the conformation of tryptophan 270A in GABAB1. This is a critical agonist binding residue. Specifically, calcium may stabilize the backbone conformation of tryptophan 270A through its interaction with the neighboring residues. Our structure also revealed the presence of novel endogenous phospholipids that are embedded within the TM domains of both GABAB subunits. Our collaborators, Dr. Oliver Fing and Dr. Tongshen from UC Davis, identified these lipids by mass spectrometry using purified calcium sensing receptor as a control. One lipid is a phosphatidylcholine and the other is a phosphatidylethanolamin. And each lipid has a 20 carbon and an 18 carbon fatty acyl chain. As shown here, we further assigned one phospholipid to each GABAB subunit based on the size difference of their head groups. PE has an ethanolamine in the head group. In contrast, PC has a larger choline group. The lipid density in the GABAB2 TM domain has a bulkier head group that can accommodate PC. Consistent with the amphipathic nature of phospholipids, the lipid binding pocket of each GABAB subunit is characterized by a hydrophilic trunk that holds the polar head and two lipophilic branches that host the hydrophobic tails. Furthermore, the trunk consists of a negatively charged patch covering the amine moiety and a positively charged area surrounding the phosphate. The lipid binding pockets are quite deep, extending from the extracellular membrane surface to the center of the TM domain. Here we compare the ligand binding locations within GPCR TM domains. Each lipid covers almost the entire range of ligand binding positions in class A, B, C, and F GPCRs. The lipid receptor interaction varies approximately 2,400 square angstroms of surface area inside each GABAB subunit. This slide shows the extensive interactions between each GABAB subunit and its phospholipid ligand. The key elements of the lipid receptor contacts are conserved in GABAB1 and GABAB2. First, the phosphate of each lipid head is anchored through interactions with conserved histidine and arginine. B2 differs slightly by also incorporating arginine 714 of ECL3 loop, thereby rendering the lipid binding pocket more electropositive than GABAB1. Second, the 20-carbon fatty acyl chain of both lipids is bent and sandwiched between a pair of aromatic residues around its turning point. A cis double bond in each fatty acyl chain forms pi interactions with the aromatic amino acids. The band is further buttressed by extensive contacts with the aliphatic part of a conserved lysine residue that lies parallel to the chain. Finally, the 18-carbon fatty acyl chain of both lipids is relatively straight, and its binding pocket is lined mostly by small aliphatic residues. This slide shows the molecular surface of each GABAB subunit. We observed lateral gaps between TM5 and TM6 of both subunits, which may serve as a potential access channel for the endogenous phospholipids. These openings are analogous to the one between TM1 and TM7 in the lipid-binding GPCR sphingosine 1-phosphate receptor. The structure of the S1P1 receptor was solved by Dr. Ray Stevens' lab. The lipids of these other GPCRs, such as the S1P1 receptor, can be easily replaced. However, the size and extensive engagement of the endogenous lipids bound to GABAB receptor suggests that they're necessary structural components of the receptor and are essential for maintaining receptor integrity and stability. We also performed alanine mutagenesis to determine the physiological relevance of the endogenous phospholipids. We mutated residues that form direct hydrogen bonds with the phosphate head group of each lipid. We identified a mutation in the ECL3 loop of GABAB2, R714A, that displayed a small gain of function. This mutation is expected to enhance the movement of PC within GABAB2 by eliminating a critical contact with the lid that covers the lipid. Our results suggest that PC molecule may act as a negative allosteric modulator of GABAB receptor by stabilizing the inactive conformation of GABAB2. In summary, we think the endogenous phospholipids may play two functional roles. First, they maintain structural integrity. Second, they serve as allosteric modulators. The combination of our previous VFT structures and current Cryo-EM data indicates that activation of GABAB receptor involves the occurrence of a sequence of events. First, agonist-induced VFT closure of GABAB1. Second, association of membrane proximal LB2 domains. Third, mechanical torsion that extends across the linker to the TM domains, and this is followed by dissociation of the intra-subunit latch between TM5 and TM3 of both subunits, and finally rearrangement of the TM heterodimer into an active conformation. structural studies by Dr. Cherizov and Gatti and Drs. Zhang and Liu indicate that the active conformation features TM6-mediated interface. To conclude, I'd like to thank everyone again for contributing to this work. I very much appreciate all the help from our collaborators, and thank you all for listening to me today.
Great, thank you very much. And it's a fantastic structure. Thank you very much for the talk today. I encourage anyone with questions, you can send them by chat. I think we can go ahead and do the, allow people to unmute. You can also raise your hand. We had a couple of questions here. All right, so users can unmute. So Ashok, if you want, you can go ahead and unmute and ask your question yourself if you'd like.
I think she had already answered my question, like asking about the effect of phospholipids and this cholesterol on this heterodimeration of this receptor. Is there any role in the heterodimeration, or it just affects?
We think the cholesterols mediate heterodimer interaction indirectly. We discovered 2 cholesterols bound at the interface, and they interact with both subunits, so we think they assist in heterodimer interaction and is in the active state. The phospholipids, again, we think that they play an essential role in maintaining structural integrity. They may also play a functional role.
Thank you.
I had a thought, just a question about that as well. So, you know, they probably play a structural role, that's for sure, but, you know, nature can make proteins without phospholipids buried in them, right? So do they play a role possibly in, you know, sort of a dynamic context, in that they give the protein some conformational access to conformational states that it wouldn't otherwise be able to access?
That's a great question, great suggestion. We didn't think of it, but I think it's very likely.
I mean, maybe I'm interpreting greasy things as more like lubricate, you know, but
yeah,
on a sort of atomic level, maybe is not true, but yeah, it's that would
great suggestion.
We had another question from Ken. Ken, do you want to unmute and ask your question? Go right ahead. Oh, he's got no mic, so I'll ask on his behalf. He was asking about a copper ion. He said, what does the copper ion do?
It's a good question. So we don't know yet. When we did the elemental analysis, we found that both calcium and copper ions were detected above background level. So we were able to more definitively identify the calcium binding site. We did observe density in the extracellular domain of GABAB1 and also close to the orthosteric agonist binding site. Some density that we believe may belong to copper ion. However, the density was not very prominent, so we cannot say with confidence that it is indeed a copper binding site. So if that site is indeed copper, then it could also be playing a modulatory role because it also coordinates ligand binding residues. But we would need to explore that further.
All right. Any other questions? Feel free to send them by chat. And I had one question. What's the current thinking about the transduction through the membrane? So I think that there's conformational changes. We've got good handles on antagonists and agonists. You know, when I think of coiled coils on the other side of the membrane, I think of them as being fairly rigid. But
so you're right that coil coils are rigid. However, these coil coils, they're linked to the end of the TM domain by a linker, flexible linker, about 15 amino acids long. And the flexibility of that linker might have caused the coil domain to be flexible and not visible in our structure. So in terms of conformational changes, so far we know that you know, the activation, the agonist binding causes rearrangement of the extracellular domain as well as the transmembrane domain. And we believe that you know, formation of the intersubunit latch maintains the inactive state, and once it's broken, it will rotate into an active conformation for signal transduction. However, as to how G protein binds and how G protein engages the heterodimer, whether it only engages one subunit or both, it's still not clear.
Great. Any other questions? I think with that, we can wrap it up. Thank you very much for a fantastic talk. And thank you everyone for attending. Be sure to catch next week. We've got Lance Westerhoff with movable type software for virtual screening. And hopefully I'll see you then. Thank you very much.
Thank you very much for inviting me.
Bye.
Bye-bye.